The green fluorescent protein, also known as GFP, is one of the greatest discoveries in modern biology. It has revolutionized biomolecular studies and medicine alike. If you ever wondered where glowing mice, rabbits, and cats came from, well, it's all due to these four men, and earned 3 of them a Nobel Prize. In 1961, Dr. Osamu Shimomura was studying a species of jellyfish known as Aquaria victoria. A Victoria had a special ability to glow bright green when threatened or agitated. After spending months collecting and processing samples of the glowing jellyfish, Shimomura finally managed to isolate a luminescent protein. However, instead of glowing green like A. victoria, the protein glowed blue. He named it aquarin, and only later did Shimomura find the small green fluorescent protein. But as it turned out, GFP needed blue light from aquarin in order to glow green, whilst aquarin needed to bind to calcium and a luciferin, a molecule that reacts with oxygen to produce light. GFP's light seemed almost effortless. It glowed with only oxygen and exposure to blue light. Shimomura continued his research, but he was largely interested in aquarin. For decades after, the green fluorescent protein went largely ignored. Doug Prasher was the first person to realise the potential of GFP in microbiology. He suggested GFP could be used to localise proteins if inserted right before the stop codon. Prasher also suggested it could be used to measure gene expression if inserted in the regulatory section. Prasher was the first person to isolate the complementary DNA for GFP, opening the door for GFP to be added to the DNA of a living organism. But soon after, Prasher's funding ran out. He applied for funding from the US National Institute of Health in Bethesda, Maryland, but the reviewer argued that his research lacked contributions to society. Prasher left the marine biology laboratory to work for the US Department of Agriculture. Now, Prasher may have been gone, but after hearing about his work, scientists from all around the world began attempting to incorporate the green fluorescent protein into a living organism. Using the complementary DNA for GFP that Prasher had discovered, Martin Chalfie began trying to add GFP to the bacteria E. coli. He attached the GFP gene to a strong promoter to create an expression plasmid that also carried antibiotic resistance to ampicillin. Adding the plasmid to a test tube with E. coli, Chalfie heat shocked the bacteria to cause it to become competent and absorb the DNA. Then plating it up onto ampicillin-coated agar, he allowed the bacteria to grow. Only those that had been transformed with GFP and antibiotic resistance grew. Applying a chemical inducer called IPTG to the bacterial dish, which activated the promoter GFP was under, Chalfie caused GFP to be expressed, and the E. coli started to glow. Soon after, Chalfie managed to incorporate GFP into C. elegans, a eukaryotic microscopic worm. This time, he had to inject the gonads of the nematode with the expression plasmid so that the offspring would inherit GFP in all of their cells. In the plasmid, GFP was linked to a specific promoter called MEC7, which is expressed in a certain type of neuron. It worked, and for the first time, GFP had been used to map a specific cell type across a multicellular organism. Another key character in this story is Roger Tsien. Tsien studied how GFP produced its light, and using this knowledge, modified it to become brighter and respond to a wider range of wavelengths. Tsien also created a palette of colours like red, purple, orange, green, blue and yellow, many of which he modified from GFP-like proteins found in corals. Having a range of colours now allowed us to track multiple different proteins at the same time. After modifying GFP and obtaining new GFP-like proteins to chase all the colours of the rainbow, Tsien had indeed found a pot of gold. He had created a vast wealth of chemical tools that have become invaluable for the investigation of biological life. Before GFP, all the previous fluorescent proteins required other molecules in order to work. And on top of that, they were phototoxic, meaning they couldn't be used in living cells. GFP could be added to cells without needing anything to be added with it, or without killing the cells. For the first time, this allowed scientists to do live cell immunofluorescence, where they could actively track the movement and folding of proteins in a cell. In 2008, Shimomura, Tsien, and Chalfie were awarded the Nobel Prize in chemistry, but unfortunately, Prasher missed out. While the green fluorescent protein first isolated from the jellyfish Aquaria victoria has brought us countless new possibilities to learn about biology and medicine, scientists still don't know why the jellyfish glows in the first place. Perhaps forever, it will remain a mystery.